I do like how sometimes you can just say the black corals because of their little like Christmas ornament crabs hanging off.
I think I can come north a bit and then Argus can lift up to avoid that.
Yep.
Does that sound good?
Yeah, I see that.
North 15?
Yeah, 10 or 15 north.
Roger. Bridge now.
I'm trying to stay at this 380 here, so I think it hooks back around to the right. So I'll be coming closer to you.
Let's do 10 meters north. Thank you.
Seeing some of the carnivorous sponges on the cliff face as well. Little individual white twigs, and I just saw a little bushy swiftia. Okay, this time to see if it's just more coral skeleton than that's what it looks like. And then right above is another nice black coral.
Yeah, Tom made a good observation. There's been a lot of dead, smaller Christmas tree corals here. There's another Asbestopluma carnivore sponge, that little white stick just above the lasers. Maybe a small paragorgia there, the red, kind of thicker stick.
Small red paragorgia. That one has its polyps out, that's why it's got kind of that fuzzy appearance. And Asbestopluma is right in the lasers right now.
What makes a carnivorous sponge different than a filter feeding sponge, Meredith?
So these are a type of glass sponge. They have these long
somewhere.
Yeah, I think 045.
Yeah, Roger.
Bridge now.
Or hooked spicules on the
Let's go 20 meters 045. Thank you.
That will catch amphipods. And then the sponge will slowly kind of overgrow and consume them. Tom is pointing out the Haliclona and Phinea sponges in the lower right-hand corner of the screen. It's interesting because it looks like the Phinea might be growing on the Haliclona. And also interesting that we haven't seen any moldy cheese sponges yet.
No.
Tom said he was thinking the same thing about the moldy cheese sponge.
Oh, and we have a skate towards the center of our screen. We have a lot of pink sea urchins here as well. More sediment on this rock face too, not as steeply as sloping.
Oh, we've got a nice little branched coral right there on the transect. We can't touch it. I think that is the pronoid of some kind, I think.
Oh, cute.
Yeah, I think it's a little Caligorgia.
That's a nice specimen. And Tom said that was a long-nosed skate that we just saw. And it looks like another Christmas tree coral in the background and another Paragorgia, the white stick up front.
Mm-hmm.
Looks like 20030.
Sure.
Bridge nav. 20 meters, bearing 030.
What's the rigidity of a Caligorgia like? Does it stand up on its own?
They do stand up on their own. They're actually quite firm. They've got a pretty rigid skeleton on the inside. And if you see another one in zoom, they've got these little rings of polyps that go around each frond.
So it's also a great view of Herc in the Argus cam if anyone's interested in flipping over to the Argus cam or the quad view so you can see both. Oh, was that a soft coral at the very bottom of the screen that we're just passing?
Looks like it.
Yeah.
It's hard, like a BAM. I don't know. It's hard to say.
Mm-hmm.
The nice little Paragorgia there.
Yeah, a nice red branched bubblegum coral. Meredith, Tom is saying something about pausing the transect.
To go look at the Calagorgia. I don't know how far back it was. Just a little ways.
Is that what you would like to do?
Yeah, let's pause and go back and take a quick look and zoom in on that guy.
Bridge nav?
There's the white coral a little ways back. Oh, no, he says no if we see another, so we're not going to pause. We're going to keep going
Oh,
forward.
got it.
Never mind.
Apologies. Just wanted to say hi.
Yeah, he says too far back, and I think it is a ways back. We would be looking for it for a while.
Yeah. But understood. If we come across one, we can consider pausing the transect then.
There's some live desmophyllum growing underneath that rock.
Oh, there we go. Those look pretty small, but that makes sense. Okay. And desmophyllum typically like the underhang of cliff faces of rocks, right?
Yeah, you'll typically find them kind of hanging upside down under overhangs on the overhanging ledges of rock.
Mm-hmm. So that's often why you'll find desmophyllum on one side of a rock versus another. They typically like to be vertical, if not slightly underneath an overhang.
More of this white paragorgia.
What am I doing here?
I think if you keep strafing northeast.
Is he still moving, or you got?
No, we're paused, and Argus is right above you.
Yeah. So. Yeah.
That's another Christmas tree coral there.
It's
That's next to the left of the white paragorgia.
Quite
It's
vertical.
a different color morph.
Oh,
We've
I
been
see
seeing
that,
the brown.
Yep.
I can go this way. So.
Welcome back, Chad. Chad's now in the control van with us. Well, with them, technically. Meredith and I are not with them. Meredith and I are in our respective offices or homes leading this dive remotely thanks to OET's magnificent telepresence capabilities.
Yep. Which is also why you occasionally get things like the barking of my dogs on in the background as I dive to the mute button.
Yeah, I think it's definitely worth saying. So we're doing a quantitative transect right now, which is a standardized method for collecting data about the seafloor. In this context, you can collect transect data and other habitat types. But we are continuing an effort that has been going on in the West Coast region for the last several years. There's a couple of initiatives that have been pushing to explore the seafloor in order to learn more about the deep sea corals and sponges that we're looking at right now, as well as other potential navigation hazards, resources, minerals within deep sea habitats. And so a lot of those same partners have come together for the West Coast Deep Sea Coral Initiative.
Yeah, seems to keep going that way, Randy.
And we've been collecting transect data like this up and down the West Coast since 2018, or supporting efforts that collect this kind of information since 2018. And the Nautilus has really been the only fieldwork we've been able to do this year because of the pandemic And so we have a lot of people tuning in and joining us. The scientists ashore in our chat room, we could not have done this without them and their help and their expertise. You'll be hearing from some of them later as watch leads. We have folks from the Northwest Fisheries Science Center, the Southwest Fisheries Science Center, and a number of different partners like the U.S. Geological Survey, the Bureau of Ocean and Energy Management. We have the other NOAA line offices with us. And we're all coming together to share our resources and our expertise and explore the deep sea, and with OET, bringing it to your homes. Tom had just pointed out in that yellow sponge there was a witch eel sticking out, quite thin and pointy. And this West Coast Deep Sea Coral Initiative will be funding the analysis of the video that we're taking and these transects that OET is helping us collect. And we, because of these standardized transects, even though we're south of Anacapa Island right now in the Channel Islands National Marine Sanctuary, we'll be able to compare the seafloor habitat and how often we saw different species of corals and sponges to other areas of the West Coast and compare them.
As part of the effort, we're also doing these collections so that we can verify what we're seeing on the transect and get clear species identifications, and also using it to build a genetic library to compare to our environmental DNA samples, which we've been taking. And we also will do with some of the species, we're doing some population genetics to find out how individual coral communities are connected to one another up and down the coast.
As good and as high definition as these cameras are, it's not always the easiest to tell what species something is on our TV screens. So the samples are really necessary to definitively say what is what.
A lot more of these dead zoanthid covered rock coral skeletons on this wall. And this one looks like it has sponges, zoanthids, and then just a little bit of living tissue left up on the upper branches.
Barely hanging on. Let's see. This cliff face has a lot of sediment on it. I wonder if some kind of sedimentation event might have killed a lot of these Christmas tree corals we're seeing. That's just skeletons now.
Oh, I think there's a pineapple siphonophore between the skeletons on the right, lower right hand of the screen.
Nice. Nice eye.
These are a siphonophore that's been thick. They have little tendrils and they float just above the seafloor and use their little tendril connections to kind of move around.
Is that a different kind of rockfish we're seeing? Do you know what species that is, Tom? the ones that are a little bit lighter in color. I know some of these are definitely thorny head rockfish, like those red guys down below.
I think some of those are split nose, is what he said above. He's looking
Split
though right
nose,
now
okay.
to see what they are.
He's the best. What a resource, Tom.
For those of you who are at home who are wondering who Tom is, Tom Leidig from NOAA Southwest Fisheries Science Center. He will be doing some watches later in this cruise, so you'll actually get to hear his voice as well. But right now he's chiming in on the science chat and helping us do fish identification as we move along through this transit. He said we can't tell due to the angle. Split nose, you're looking for their split nose. We've got more dozen film there on the wall next to that coral skeleton.
Yeah, that's more of the live kind of skeleton that we just collected the dead skeleton of. So I guess that satisfies my itch somewhat. Tom is also letting us know that he's also seen aurora, split nose, and blackgill rockfish so far as well. So we are coming across a greater diversity of rockfish at this depth, which is roughly 380 meters. So I think, Meredith, you were right. These look like the rockfish I was asking about earlier, and Tom is saying these are split nose rockfish.
An interesting kind of channel looks like we're here that's more sediment filled.
Like a slide might have happened.
Yeah, like a little slide zone.
With a tall stick. Oh, that's the dead Christmas tree coral, I think. We have one of those Gallophyliid crabs.
Yep. Yeah, somewhere in there, I think. Looks like a piece of cement.
Yeah. It's very angular.
More paragorgia there in the cliff wall. How far have we come in this transit?
This is about 110 meters,
Okay.
120.
And we're still at the same depth at which we started, 382 meters.
Roger.
And crowding along the water.
Another objective that we'll have on this dive, this is our first dive within the sanctuary. Oh, is that clavularia area on the left?
On the left. Yeah, it's a little bit of clavularia on the left. In the rocks
It's a
there.
soft coral.
Clavularia is, yeah, it's a coral that grows in a flat in the stoloniferous form over the rocks.
Can you describe to me what stoloniferous means?
Oh, we've got an octopus tucked down in that hole behind those sponges and coral.
Yeah.
Oh,
that
good
yours?
eye.
Yeah. Think.
It is wet
So
down there.
stoloniferous is a growth form where they grow on like a rock flat surface, and they send out these little membranes of tissue that then grow more polyps but flat on the rock.
Got it. Cool.
They're more encrusting.
Another project of the West Coast Deep Sea Coral Initiative that will be supporting the analysis of these transect videos is we're creating a deep sea coral portal. Oh wow, you can see the octopus and his tentacles. Oh, that's a good shot. We're creating this portal that has a lot of spatial information in it, and we're using actual deep sea coral and sponge observation records from dives like these, from museum collections, from trawl surveys for fish where corals are caught on accident. And students will be able to interact with those observations within a map. And we were working with this data from the Deep Sea Coral Research and Technology Program's national database. And we were trying to group them into more accessible categories, and stoloniferous was a category. And I found myself not being familiar as easily with the biology and what the stoloniferous form meant. And I could not get it wasn't obvious just from Googling it. So that was really helpful.
There's a paper by Kathy McFadden and co-authors that actually has a really good illustration, like drawn illustration of a stoloniferous coral, where you can really see what they mean by stolen.
There's another octopus hiding in the sand.
Mm-hmm.
Is that what that is? I could
Yeah.
not tell what that
Really?
orange thing was. I figured it was a sponge. Great eye. And another gully where it looks like a lot of sediment has been shifting down the cliff side over the past, well, who knows how long, but for a while probably. More urchins that we tend to see in pretty sedimented areas, but they can be found on rocky surfaces as well. Another small paragorgia there, that white, thicker, stick-like feature. And a Christmas tree coral with more Galatheid crabs on it. Well, at least one.
Bridge now.
Got a nice side-by-side of the two different types of Galatheid right there.
Oh yeah.
20 meters 015.
In other words, these impressively steep but yet still sedimented slopes.
Mm-hmm. Where there's a lot of sediment and a lot of sediment movement, you won't find a lot of corals and sponges because they are filter feeders. And if they get smothered by sediment or they're not able to feed because there's a lot of sediment movement and they're essentially clogged by the particles, that will essentially smother them. And sometimes there's sedimentation events versus more consistent high flow areas where a cliffside can give way and it can fall kind of like an underwater avalanche. And that can kind of be a mortality event, which I'm wondering if that's why we were seeing so many dead Christmas tree corals earlier, if such a sediment event could have happened and just wiped it all out, all shapes and sizes, so different age groups. But that is just a guess. Oh, what's that flatfish in the center? Is that it's not a tonguefish. It's a cute little orangey flatfish.
I'm not
I'm
seeing
sure.
a cute little orangey flatfish.
It was pretty well hidden. Oh, Thomas named over sole. It must have just been really small. Thomas saying, well, they all start out small, which is
Bridge
true.
nav.
Can we
You can hold position there and add a step 20 meters 060.
pop and left? That's another Caligorgia.
I think you cut out a bit. Did you say you wanted to pause?
It's not now.
Okay.
Yeah, I got a good look at it as we kind of drifted closer. We have another octopus.
Yeah.
Oh yeah. It's looking like a crab.
I like how it's stealthily kind of creeping along the bottom. If I move really slow, they won't notice.
What's that?
Sponge.
Nice little Christmas tree. Oh yeah, and a big, tall barrel sponge there. I always love how those barrels get so filthy on the outside, and then you can tell they're alive because they stay perfectly white on the inside.
Bridge nav. Can you call that movement again? It doesn't seem to be moving. Thank you.
Yeah, I think it's interesting, Meredith, that some sponges look clean on the inside and outside, but then you have some, like the one we just passed, that just, they're so covered in sediment.
Yeah, and other types of sponges, that would be a sign that they're dead, but on those barrel sponges, you'll have to look at the inside. And when you get those on the deck, they tend to have these really long spicules that help catch all that material. There's an interesting coral hanging upside down there.
It fainted. So scared of heights. That's
Bonus skate. That seems to be hunting that Galatheid that's running for its life.
Sorry about that. It's just starting to move now.
All right.
Right at the end of your leash, I think.
I appear to be at the end of my leash.
How much farther do we have in the transect?
So we've done 150 line of sight, but we squiggled a bit along it, so probably closer to 160, 170. Yeah, I'd say 170.
Roger that. Thank you.
A little Swiftia on that boulder. Another pink paragorgia.
Oh yeah, yeah. The Swifty is the smaller red, really finely branched coral, and then the Paragorge is the chubbier coral.
Yeah, the Swiftia often on the screen just looks like kind of red fuzz.
Yeah,
Bridge nav.
gotta have a trained eye for that.
Let's add one more step there, 20060. Thank you. Just hop over it. That ought to do
I
it.
haven't seen many more Asbestipluma carnivorous sponges, except back at that area that we were at earlier.
It's gotten a little more sedimented. I wonder if that's why.
Yeah, that's true. We have seen a lot more sediment here.
Oh, there's one. We spoke
Hello.
too soon.
There we are.
We're finishing up its transect, and very shortly, watch changes are going to begin. We'll be changing out ROV teams and data loggers, and then Lizzie and I will be jumping off the headsets when the transect is done to turn it over to Tom and Lisa and possibly Abby.
Yeah, Lisa is another sanctuary person like myself. She works for the West Coast Regional Office, and Tom is the person we've been referencing a lot for fish IDs who works for the Southwest Fisheries Science Center. You can see that coral skeleton in the middle is covered with the invertebrate. They're called zoanthids. So although it kind of looks like a coral with a bunch of polyps, those are actually a different species of invertebrate, and it's not the coral itself.
Sorry, Jess, are you all good? Do you need me to let you know where we're at?
Got a nice soft sponge there.
The big white one.
Mm-hmm.
It's so hard to tell what's a stalked sponge and what's a sponge growing on a dead coral sometimes.
So it looks like Tom is proposing, and possibly since the ROV crew is going ahead and switching out now, that we might go ahead and tag out our teams as well.
Absolutely, it's been a pleasure. Nice working with you again, Meredith, although be it remotely.
Yeah. Pleasure running a dive with you, and I look forward to more. And this is a
To midnight.
great way to start off. Yes, midnight tonight. Start off
Oh,
working in
wait.
the Channel Islands.
Yes, wait, that's true. Sorry, I'm forgetting my schedule. So we are back on for a launch at midnight, and we will be looking at a hopefully hard bottom feature habitat, actually not too far from here, which is South Anacapa, and near the footprint reef. So we're hoping to find some great deep sea corals and sponges there. We're going to start at 800 meters and work our way up. So I look forward to it.
Yep.
Hello, folks.
All right.
So it looks
Hello
like
and goodbye.
so it looks like we've come 180 meters for this transect. So we want to keep going and finish off that last 20 meters, or just mark it.
I think we can just continue on for the last 20 meters and then decide where to go from there. We're
Awesome.
going to do a switch, watch switch really quick, but yeah, I think we can just finish this out. Shouldn't take too much longer.
Roger.
Thank you. You guys have a good dive.
Thanks.
Thank you.
Yeah.
Hey, Jess, I'm going to call in 10 meters 060 to continue.
Yeah,
Is that
sure.
good?
Yeah.
That's your standard box.
Roger that. Thank you, Kim.
So I'm looking to 060.
Roger. Thank you.
Yep, we can hear you.
I can't hear.
Yeah.
Sorry.
Yeah, SPL is great.
Okay, so hi, this is Lisa on SPL and Nautilus.
Hey.
Hey, Lisa.
Welcome, Lisa.
Hey, hi. So there's no echo, everything's good?
Yeah, you sound pretty good.
Great, great. We're just waiting for Tom, and then we'll do some introductions.
Perfect.
Right, and I'll do another 10 meters, but this is 040 to keep us a little closer.
Yep, keep us
Great.
along the contour. Great.
Mm,
Yeah. Thanks, Kim.
Yeah.
I think I see some line here. Gonna change my heading.
Oh, yeah. Good eye.
Looks like we're clear.
It also looks like we've come to the end of this transect.
Great.
200 meters, so. Is that good to mark it? Science?
Are we good to mark the end of the transect?
Yes, this is Lisa. You can enter it into the data log and I'll enter it into the tracking sheet. Thank you.
And this is T5?
Yes.
Awesome. Thank
Yes,
you,
this
Mary.
is T5.
Okay, great. Yeah, so if you guys want to go over the next destination here, because we are a little south of that waypoint AJ.
Okay.
And then, so we can either continue along this cliff feature and then head up to AL or AK. I think.
Let me just, can we just wait until Tom is able to log in? If he isn't in 5 minutes, then I'll figure it out.
Okay, great.
Does that sound okay? Good.
Yeah,
Thanks.
so we can just continue along the contour for now.
Yeah, Tom's login should
Yeah.
be available now,
Great.
but Tom, let us know if you're still having issues.
Looks like another either rocky feature or something that line snagged on there. If anyone will hop us forward a little bit on this heading so we can get past this line.
Yeah, I'm going to put in 20 meters, 040. Is that good? Or I can keep it down to 10.
Yeah, 040 is fine. 20 meters, good.
Great, thanks. Yeah, I assume the plan is to keep along this contour till AK. Yeah.
I believe so. The dive is supposed to end with the ROVs on deck at 8 o'clock this evening, so at the end of this watch.
Yep. And I think it
Yeah.
will only take us about 30 minutes to get up from the 350 meters or so.
So if Tom's listening, we can see his login going in and then getting logged off, going in and getting logged off. So I don't think it's anyone else. I think it is free, but perhaps Tom's having problems getting into V-Link right now.
I had some problems with turning the SPL talk function off. It would log me out, but it's working now.
Yeah, if one of you wants to message him, unfortunately I don't have access to the science chat, but you know, sometimes the old what do we call it? System
Do one more
system
move.
cycling, where you just turn it off, turn it back on again, just restart it. I don't know if that would help on his end.
Yeah. He wants the van number. And I'm trying to find it right now. Oh, here, it's under key contacts.
Probably makes sense until we hear otherwise to follow that contour around.
Yeah, and then why don't we do a little round of introductions? I've been itching to do this because this is my first Nautilus dive, first time I'm participating with you all.
That's right, Lisa.
So can
Woohoo!
we? Yeah. So I guess the way that it works is we start with the back, no, the front row. The front row. I'm the back row.
All right, we'll start
hear
at nav. How about that?
Oh yeah, so
Yeah.
yeah, so I'm Ben Gracian and I'm sitting in the navigator's seat and we also do the mapping. Yeah.
Cool.
Jess Sandoval, sitting in a Herc seat, ROV team.
Hello, I'm Kim Wallace, ROV team, sitting in the Argus seat. And Marley's on the phone, trying to sort out technical difficulties.
Is Marley video then?
Yes.
Or is she data logger? Okay, she's video.
Oh.
And who's our, sorry.
Oh, you're good. I'm Mary Dury. I'm the data logger.
Hey, Mary.
And I'm sitting next to Mary in the back row. I'm Chad King from Monterey Bay National Marine Sanctuary. And we've been hijacked. I'm just kidding. Weather was really bad in Monterey Bay Sanctuary, so our colleagues down south are using the time to get a little ahead on their cruise. And so we're diving today and tomorrow in the Channel Islands National Marine Sanctuary.
Yeah, because he's still at 045, so I'll just keep us at their 035 10-meter move. We're so shallow, it's responsive. Does that sound good?
Sounds great. 035.
All right.
He's going to move us a little closer
Okay.
to that depth contour, like you
Yeah.
suggested, in 035.
So those of you who are listening in, you just heard the introductions from the ocean exploration team who operate the Nautilus. And we are currently diving in Channel Islands off of the Anacapa slope to the southeast.
Roger.
And my name is Lisa Vonik. I work for the Office of
Okay.
National
No
Marine
worries.
Sanctuaries, like Chad. And I'm a policy coordinator. I am trained as a scientist, but I haven't been really doing science actively like Chad and a lot of other folks at sanctuaries for at least 15 years. But it's really exciting for me to be back in this chair again and to be on this Nautilus cruise in the Channel Islands. And we're waiting for my watchly Tom Leidig to join. And I think I'm hearing in the science chat that we could take a Niston the next time we sample, okay? And we're on our way to AK along this contour line, correct?
Yeah, so we're just following the contour for now. We're gonna have to get a little bit closer to it, to the cliff. But yeah, if that works for you, that seems great to us. It's just this little ditch of a feature that we'll get around and hug tightly and get up to 8K.
Okay, sounds good. And then from AK, we're gonna go up to AL or AM, but it just depends on what we
Yeah, wait, that
find
were
along
you
the
guys
way.
decide. Both of those will be,
So
AL will just be upslope to the ridge, and AM
right,
will be along the contour.
right. And so how far are we from AK?
Let's see. We're about 800 meters, 800 meters.
Okay. So we could probably get in another transit, a time transit.
Yeah, for sure. This may be a little tricky along this contour with the cliff. So
What works best for you? What would you prefer to do?
Yeah, so we can lateral along this contour, and best thing is just the distance, and we'll try to keep a reasonable speed and speed over ground. Yep, that works for us.
awesome. Yeah, I'll just try to get Argus a little closer.
Yeah, so maybe if we go maybe like
020 or
020.
north. Okay.
Be great. Thanks, Ben.
Hey, this is Tom. Can you guys hear me?
Yes, we can.
Yes,
Hello, Tom.
Tom, welcome.
Hey, Tom. I made it on.
Mr. Leidig.
How are we all doing?
Good. So we went through a round of introductions, and so now it's your turn.
Oh, great. Let's see. I'm Tom Leidig. I work for the National Marine Fisheries Service in Santa Cruz, and I study deep sea corals and fishes. I don't know what else do you want. I'm sitting here.
Well, I'll fill in. Tom is really our local regional fish expert, and he is the one who leads up a quite dynamic and well learned team of folks who analyze all of the data that we collect via video. So he really knows what we're observing while we're traveling along the bottom here.
Oh, great. Now everybody's going to believe that.
Well, just fake it like you always do. So maybe I should let Tom know that our Herc pilot is Jessica, and Kim is the Argus pilot, and Ben is our navigator. Marley's on video, and Mary is our data logger.
Hi, everybody.
Thank you.
Hey.
You're on it, Lisa. Good job.